All right, welcome back. Today we're going to do topic 2, which is all about microscopes, microscopy, which is a fun word that you can tell your friends. This will not be as long as topic 1. We should be able to do the whole thing in one video. We're going to talk about what the parts of a microscope are, how you can use them, and then I'm going to post a link to a really good microscope simulator, and I'll walk you through using it. All right. What might we need microscopes for, and how might they help us in science? Go ahead and write down some ideas there. Essentially, a microscope, micro, means small. And the scope is the thing we use to see. So we use a microscope to see things that are microscopic, very, very small. These are pictures of different microscopes. This is an older sort of microscope. This is the microscope that very similar to the microscope that you might be using if you are currently in Science 10 at Center High Campus. We use these to see very small things. What are some small things that you might see? What are some small things you might want to look at? Well, let me tell you. We just spent a whole topic talking about cells. Cells. You know, there's other things too. Look at the pixels on your phone. That's pretty cool. You might look at inorganic things as well as organic things. You can use a microscope to see very small crystal structures, and that's pretty cool. But for biology, we're going to use them to look at cells. Everything we've said, all living things, not everything, but all biotic things are made of cells. How can we prove that? Well, let's take a look. Some history. So Zacharias Janssen and his father created the first compound microscope. A compound microscope is a microscope that's going to use several lenses. We talked earlier in the first lesson about Anton van Leeuwenhoek and his advances to microscope technology, but the first microscope wasn't made by him. And the first microscope could magnify objects 4 times their real size. So whatever you're trying to look at would look 4 times larger under the microscope, which is still significant. That's 4 times more than your eye could see. But in order to see the very small cells that we're going to be wanting to look at, we need something more powerful. In biology, we used 2 types of microscopes. We use light microscopes, which sometimes we are going to refer to as our compound microscopes. That's not 2 C's, that's a bracket, but I'll make that more obvious. Compound microscopes. And we use electron microscopes. We use light microscopes and we use electron microscopes. This is the one that you will use in class. Unfortunately, we don't have an electron microscope at Center High that you can use, although that would be pretty cool. A light microscope is going to use glass lenses to magnify. Similar to the first microscopes, it's just an improvement on it as we get better with lens technology. You can use different types of lenses to get a closer picture. It's also going to use light from the bottom to show through. Because you're using light from the bottom, you can see color. So the advantages to a light microscope is that you can see color but the image is flat. You can see color, but the image is flat. Another advantage, though, is that the specimen, whatever you're looking at, remains alive. You can contrast that to an electron microscope. The electron microscope is going to use electron beams. If you think back to the chemistry unit, electrons are very, very small negative particles. You're going to fire a bunch of those very small negative particles through this tube. It's going to hit your specimen. It will bounce off, and then it will hit a detector all around. As it hits the detector all around, you will get an image that the computer will build based on how the electrons bounced off. The result is something like this. You get a really high resolution image. And it's going to be a three-dimensional image. You get a high resolution 3D image. These are not pictures of the same thing. On the left here, we have some plant cells, and on the right, we have some pollen grains. This is a super high resolution 3D image. The cons of using an electron microscope are that you have no color, and if you are looking at a living specimen, when you bombard it with the electron beam, it will die. It will kill living specimens. It's also way more expensive to use an electron microscope than a light microscope. A compound microscope, don't think of this as being another type of microscope. Put the word light above compound microscope. The light microscopes that we use are compound microscopes. It's called a compound microscope because you have a combination of lenses to magnify objects. There's going to be two lenses. There is the objective lens. I'm going to use blue for objective lens. There is the objective lens. Think object. It's the lens that is closest to the object you're looking at. And then there is the ocular lens, where you look through. Ocular, think oculus, think eyes. It is the lens that you are going to, this is the one that you look through, and this is the one that is closer to the object. Take a look at this diagram here. There is a bunch of labels at the bottom of this page that will tell you what each thing is. I'm not going to go back and forth, but I'll label these, and then you can read at the bottom what each one is. Number one, we'll just go down in numerical order. Number one, this is called the body tube, and you should know the parts of the microscope. This is the body tube. The body tube here, this is going to support the eyepiece. Now remember, these definitions are just at the bottom of the page, so you can read those. This here is our revolving nose piece. Hard to get in that box there. The revolving nose piece is going to allow you to select which objective lens you're going to be using. Here we have 3 different objective lenses. Now I'm going to decide that this one, the first one, which is number 3, is going to be our lowest power objective. On this picture, it's hard to tell which is smaller between this one and this one, so I'm going to pick that one. This is our low power objective. And I'm just going to write beside it, it's the smallest tube. The next one is going to be our medium power objective. And then our last one right here, this is what number 5 is pointing to. That is our high power objective. And it is the biggest objective lens on the nose piece. It will have the greatest magnification. Number 6 is pointing to these two metal clips on the stage. These are simply called the stage clips, and they will attach whatever your specimen is to the stage so that you can look at it. Underneath here, this is one that a lot of people forget about. This is called the diaphragm. Sometimes you might see the word iris diaphragm because it's similar to your iris, or the colored part of your eye, because it can open and close to change how much light comes in. You might see on a quiz the word iris next to the diaphragm. This piece down here controls how much light comes in and out. And the source of our light is on the bottom here. This is our light source. Moving back up to the top, this is where you would actually look through on this microscope. This is our ocular lens. This next piece here that kind of supports the whole lens structure, this is called the arm. The piece where you put your specimen on top of, this is your stage. Then on the side here we have two knobs. Sometimes these knobs are separate like they are right here. Sometimes they're stacked on top of each other. So actually this knob will be on top in the middle of this knob, but the larger knob is called the coarse focus knob, or coarse focus adjustment is what's written at the bottom of the page. And the smaller one is the fine focus knob. And then finally, at the very bottom, this is our base. And altogether, these are the parts of the microscope. Every microscope is going to look slightly different. Some microscopes are going to have two ocular lenses that you can look through both eyes. Some microscopes, again, will have this fine focus knob on top of the coarse focus knob, but they're going to have the same basic parts that you should be able to recognize and know the function of. The whole purpose of a microscope is for magnification. Magnification is the process of making something appear larger. Some great writing, Kyle. Appear larger. To get the magnification of our compound microscopes, we multiply the power of the ocular lens by the power of the objective lens. I'm going to use red for the ocular lens and blue for the objective lens. So let's say the ocular lens had a 10 times magnification and the objective lens had the 40 times magnification. Our total magnification would be 10 times 40, which is 400 times. So if you were looking through a microscope that had a 10 times power ocular lens and a 40 times power objective lens, the object would look 400 times bigger. Another word that you're going to see is this idea of resolution. Resolution is the degree of clarity of an image or an object. So if I pull up a YouTube video, which is just right here, and you can do this to this own YouTube video. On YouTube, this is referred to as quality on the bottom here. This is another way of thinking about it is resolution. So 1080 HD, that's high resolution. You can see everything is very clear. But if I change that back to 144p, everything is pretty blurry. That is a low resolution. On the previous page, I talked about how, or a couple pages back, we talked about how an electron microscope has a high resolution. So it's a very clear image. You can pick out a lot of detail in this image. It's a high resolution, whereas this one is a slightly lower resolution. It's kind of blurry around some of these chloroplasts in this plant cell. Okay, we're going to calculate the total magnification of the following lenses. Now, they use the words scanning objective and low power objective. I'm just going to cross out low power, and I'm going to write medium power objective, and scanning will be my low power objective. You have to look at what's actually written on the side here. Now, speaking of resolution, not the most clear pictures, but this ocular lens has a magnification of 10 times. And this medium power objective has a magnification of also 10 times. So if they both have a magnification of 10 times, our total magnification will be 100 times. Our ocular lens is the same in all of these with a 10 times magnification. Our low power objective lens has a 4 times magnification. That's what this 4 is referring to here. And our high power objective has a 40 times magnification. That means that the total magnification here would be 40 times looking under the low power lens and 400 times looking under the high power lens. An important piece to know is how do you look after a microscope safely? Microscopes are expensive pieces of equipment, and it's full of glass and very specifically fine-tuned lenses that are easy to break. So it's important to know how to take care of them. First of all, always hold them straight up and down. Don't tilt them on the side. Don't hold them upside down. Place one hand on the base of the microscope when you're carrying it, and the other hand on the arm. You could reverse which one's going in those blanks, but the important thing here is that you're carrying the microscope with 2 hands. All parts should move easily. Do not force them. Do not attempt any repairs, all that business. Only clean the lenses with lens paper. The only acceptable method of cleaning lenses is with lens paper. Don't clean it with a paper towel. Don't clean it with chem wipes. Clean it with lens paper. Otherwise, you'll scratch the lens and then you will ruin the clarity of the image. After you're finished with the microscope, turn it off and unplug it. And lower the stage completely using the coarse adjustment knob. Oops. And leave it on the lowest power lens. Another piece that's really important that isn't written here, but it is written on the next page, is that we should always begin viewing every slide using the lower power lens and then focus using the coarse adjustment knob on the low power lens. Once you get off of the low power lens, then just use the fine adjustment knob. So you can use the coarse adjustment knob first, and then you can use the fine focus knob. When you're on a higher power lens, only use the fine focus. That's the big thing here. Never use the coarse adjustment knob when you're on the higher powered objective. I would even say never use the coarse adjustment knob once you're past your lowest power. Okay, this is part of a lab. which you might have done in class, but if you are not currently enrolled in a physical class, instead you're taking this through InReach, or if you just want more practice, I've posted this microscope simulator. It's quite good. I'm going on the Explore option. You can click the Learn and it will help you out as well. But just to give you a demonstration, so we're going to look at a plant cell here. So we're going to take a cell. It's now on our microscope stage. So if I zoom, I don't know if I can make this page a little bit larger. That's better. Okay, perfect. This is the microscope slide right here. You can see that you can't really see anything. The first thing I'm going to do is I'm going to change my coarse focus. This is what I'm viewing through the ocular lens right now. You'll notice that on the actual microscope simulator, as I move this back and forth, the stage moves up and down. Because it moves so fast on the coarse focus, I don't want to use this with the longer lenses, because then I could push the stage into the lenses. I'll move this until the picture is pretty well focused, and then I'll switch to the fine focus until I get a really nice clear picture. I also have the iris diaphragm, which is on the bottom here for this microscope, which will change how much light goes in and out of this image. Under this low power, you'll see that a lot of light really washes out the image. So I'm going to lower the light so I get the clearest picture of these plant cells. Once I switch lenses, it should be pretty well focused, but I can still use the fine focus here to get a better image of these plant cells. As I move up my magnification, then I'll have to let more light in so that I can get a better view of what I'm actually looking at here. Say that that's pretty, I want it to end there. There we go. This is some plant cells here. This is the nucleus where I have my mouse on here, and here are the chloroplasts. You can't readily see the other organelles. You have to apply a stain in order to make more contrast to see many organelles. So not all organelles are going to be visible. Our microscopes don't have a 100 times objective lens. And keep in mind here that this ocular lens is still 10 times. So although I'm using a 40 times objective lens, the actual image that I'm looking at is actually 400 times greater. If I'm about to use the 100 times, which we don't have in our labs, you actually have to use oil to help with the, because the lens is pretty much touching, and then also to help with the focusing, just with the refraction of the air. So if I change how much light's going through, this is what this would look like under a 100 times objective lens, which if my ocular lens is 10 times, this is 1,000 times greater of a picture. Once I remove the slide, it's important to clean it, again using lens paper. And then we go back to our lowest objective lens and lower the microscope stage. I'd encourage you to play around with the simulator. It will give you a good overview of microscopes, especially if you didn't get a chance to attend the lab. And in the next video, we'll start talking about cell membranes.